Feeling good, wanna feel great. Longevity starts, don't hesitate. Too much noise, what do you choose? Trendy hacks, conflicting news. Skip the myths, come hear what's true. From world-class experts straight to you. First and third Thursdays, 7 p.m. Science, health, and our team. Brain to heart, from food to rest. Live the talk on living your best. At 7, we'll see you there.
When a yeast divides, it must create membrane damage when you have a separation of the membrane between the mother and the daughter cell. So is this pathway involved in cell cycle in that sense?
Again, Brian, it's an excellent point. The answer is yes. Actually, yeast cytokinesis in wound healing is very similar response. So the regulatory molecules are overlapping quite significantly. And also at the bud scar, we do see the local externalization of phosphatidylserine in very similar, analogous to what we see at the laser damage site.
Welcome to the National University of Singapore Yonglulin School of Medicine Healthy Longevity webinar. Thank you for joining us today. You just saw a snapshot from our last episode with Associate Professor Kaiko Kono about using yeast to study membrane damage and aging, of course. You can watch the full episode on our YouTube channel. Today, we focus on computational biology. Professor Marija Cvijovic is with us. Please send your questions and comments while using the Q&A function. And these will be perfectly moderated by Grace Key, our research assistant, and she will introduce herself first.
This is Grace. She is a research assistant who studies aging, which is bold because the stress from this project is definitely aging her faster.
Whoa, that's rude. But okay, fair enough.
Grace works with Professor Brian Kennedy and Professor Esther Wong, and she's trying to find drugs that activate autophagy, so all of us can age a little bit slower. But Grace, what is autophagy?
So autophagy means self-eating, but don't panic, it's just the cell's clearance system. The cell packs its junk into vesicles called the autophagosome, and you can think of it as loading up a rubbish truck. It then sends the truck to the lysosome, which acts as an incinerator to break down and recycle all of the junk. So in short, autophagy is the cell's quality control system.
But with age, this system becomes slow and inefficient. It's like the cell tries to do spring cleaning, but the mop is broken, the broom is missing, and the lysosome has quit.
And junk builds up. Disordered proteins and damaged components will disrupt normal function. This contributes to age-related diseases, especially neurodegenerative diseases like Alzheimer's and Parkinson's.
So how do we fix that?
With TFAB, transcription factor EV, think of it as the Marie Kondo of your cell. It's the one that says, let's throw away everything that doesn't spark joy.
TFAB doesn't just clean, it multiplies the cleaning staff by upregulating autophagy genes and lysosome production.
If we activate TFAB, we can restore autophagy and slow down age-related decline.
So Grace asked the ultimate question. How do we activate TFAB efficiently and safely?
Through drug repurposing. FDA-approved drugs already have known safety profiles and mechanisms, which means we can bypass years of early-stage testing. This also means better translatability.
In other words, Grace is trying to cheat the system, but legally.
Strategically.
And so she screened almost 3,000 drugs from the FDA-approved and pharmacopeal drug library to find compounds that activate TFAB and autophagy in hopes to find the next possible longevity drug. She expected some supplements, metabolic modulators, maybe something boring, but she did not expect this.
Antipsychotics, one of the most prominent groups that activated TFAB and boosted autophagy in my screen.
cause confusion, concern, a tiny identity crisis.
But antipsychotics can cross the blood-brain barrier, which means it can activate autophagy not only in the body, but also in the brain.
She also realized that antipsychotics may help address a new hallmark of aging, psychosocial isolation.
As we age, physical decline comes with psychiatric changes like anxiety or changes in sleeping patterns. So together, this can lead to withdrawal and psychosocial isolation.
So you're hoping antipsychotics can help support both the physical and psychosocial aspects of aging.
Yes, exactly.
And which antipsychotic are you looking at?
It's called Sertindol, which is originally used for schizophrenia. So interestingly, schizophrenia patients show accelerated aging and autophagy deficits. So naturally I thought maybe with careful dose optimization, could these drugs then be used to help boost autophagy in healthy individuals?
Grace, are you sure about this?
Look, no one expected rapamycin, an immunosuppressant, to be the poster child of longevity research, right?
Okay, point taken. And outside the lab, Grace is also the vice chair and secretary of the Young Scientist Club. Along with her team, they've organized countless events focusing on scientific development, professional development, and mental well-being.
I really enjoy building a supportive community for young scientists because it makes research work feel less lonely.
And that is Grace. She repurposes FDA-approved drugs, activates T, Fab, and autophagy the Marie Kondo way, and tackles aging at the cellular level, so your cells stay clean and functional.
If you'd like to know more about my work or the Young Scientist Club, you can find me on LinkedIn. I'll also be moderating the Q&A later, so remember to put your questions down in the chat below.
See you, Grace.
Wonderful, Grace. I think from self-eating to antipsychotics. So, wow. Good. We are changing gears a little bit. Our guest today is Professor Marija Cvijovic, who is a computational biologist from the University of Gothenburg and the University of Technology in Sweden. And she leads research that brings together mathematics, biology, and very big data to understand aging and, of course, longevity. She also is internationally promoting cross-disciplinary work in computational biology. Marija, it's our really honor to have you here and really making sense, hopefully, out of big data. The floor is yours.
Thank you, Andrea. Thanks for the invitation. I will jump straight to the topics that are very close to my heart and everyone's heart, I guess, here at the Healthy Longevity team. So the how to open the aging talk is basically how we age. And as you probably understand, we have thousands of definitions of aging. And coming from the theoretical math background, you usually search for a source for the definition. And you can go to the dictionary, which will tell you that this is a process of growing older or showing the effects of increasing age. Many of us would not like that. Maybe the second definition of acquiring desirable qualities by being left undisturbed for some time sounds a little bit better. But it's simply the process of growing old. If you ask me as a mathematician, we have a very brutal definition of what the aging is. And that it's on a statistical level that simply the likelihood of you being dead is increasing with age. Very correct, but doesn't really tells us a lot. And this is the question that basically puzzles a lot of people. And one of my favorite definitions in aging from all of this, what I have read, is basically the quote of Bob Hope, who tells you that you know you are getting old when the candles cost more than the cake. And that's probably the most accurate description of aging that I have ever encountered. But how we are working with aging, what is it that we are doing? It's trying to basically ask right questions. many times, being a mathematician, people usually think that I'm adding numbers, solving problems, but actually most of my time I'm trying to pose a right question that I can answer. And that's very nicely summarized in the famous John Takis. He's a famous statistician who said that looking for an approximate answer to the right question is probably way better than finding a precise answer to the wrong question. And that's exactly what we are trying to do with aging. But we are not the first. I mean, the history taught us a lot of lessons, and I will quickly recap what is it that we have learned so far. And the aging model started long time ago, 1825. Gompertz developed the survival models of aging that are very similar to the statistical model that I told you about. Then the field moved to a bit more complex age structured models that use so-called Leslie matrices to divide the structure, to make a structure of the aging population. And then the field was a little bit quiet when it comes to the computational approaches until the 90s, when this big network model came. It was probably the largest model developed by Kirkwood and Kowald in 90s, but also showed us a lot of issues that you can go big with math, but biology doesn't like it. There's still a lot of level of complexity that we could not bridge with the complex math, and the complex math could not be solved at that time either. So what happened that people decided to go small again. We started deriving models on the small processes, isolated processes, looking at the kinetics, looking at the measurements that we could have experimentally confirmed. Then that worked pretty well, but being humans, we always want to go bigger. So we tried to go in a direction of genome scale models, where we don't need a lot of experimental data to build computational models, but it's only based on our observations. And then this led to the huge breakthrough of all the experimental techniques that enable development of a lot of data, particularly on the personal omics, that at the end led that we are now able to analyze a lot of this data, and particularly with machine learning. But still looking at this history and looking where we are now, the answer is not always in the data. And I like to use this analogy with the Formula One, where you have like 200 sensors on a Formula One car that you continuously measure many different components of the car and analyze it in real time. But despite all of these efforts, we still manage to fail. So the Formula One cars will crash during the race, and we have no idea why this is happening. So this basically brings us to the two kind of main points here, that we can measure a lot of things, but we cannot predict the outcomes. And to put the complexity even further, we have built Formula One car. We know every single mechanism behind the car, and we still cannot predict the trajectories of it, despite measuring so many data. So now, coming back to us, we have a lot of wearable electronics. We are capable of measuring hundreds of biological parameters of an individual. And on top of the complexity that we have, we didn't build ourselves. We don't have a blueprint. We have no idea what is really happening inside. So you can imagine that we are trying to reverse engineer a system that it's adaptive, evolving, extremely dynamic, and in the language of Formula One is moving 300 kilometers per hour. And how we are going to do this? So this is where the focus of my research is mainly about, and I will tell you how we use computational biology and to understand the performance of the system. And the best analogy to this is also the old Indian legend that says if you put a blind man touching the elephant, they might identify different things, but they will not understand the system as a whole. And you can imagine in health that we have all these data points as an isolated systems, but the health as such is a system, and it's a whole system that we have to analyze. And the field where I work, systems biology, lives basically in the space between these two and bridging the measurements with the meaning. And the organisms that I work is yeast, similar to the previous session that last time that there was this seminar. And the reasons for this are rather simple. I come from a math field. When I entered biology, I needed something simple to understand. I am in Gothenburg, that it's like the hub for the yeast systems biology, where there are many discoveries on yeast aging have been made, and that are very significant ones. So it was very natural that we basically start building our technologies and our methodologies in yeast. And I will guide you now to what things have we done in the past years. We started by developing mathematical models of different processes in yeast on different scales, starting from the damage accumulation, growth, and division. That is a small kinetic models and tells you how this yeast cell is dividing, what is happening inside when it comes to damage. Then we moved to nutrient signaling, trying to understand how yeast is sensing different signals, what is happening in the yeast cell during this process. And we tried also to go very small with the kinetic models, measuring certain parameters, but also went very big and look at the whole nutrient signaling network in yeast using the Boolean modeling, which is like kind of on and off switches that you can imagine. And then we also look at a very important aspect, that's metabolism. And metabolism is typically studied using flux balance analysis, so-called FBA models, and you build this like genome scale models of metabolism, and they're very robust and very easy to predict outcomes of it. So after years of building those small modules, we were thinking, why don't we try to make a plug and play kind of platform and put these models together and build basically a big model of yeast replicative aging. And after many failures and after many years, we managed to do that eventually and build so-called yeast multi-scale model of aging that has 226 components. It's the largest yeast model, aging yeast models existing so far. And what is also important that it bridges very different temporal scales that are happening during the aging process. Some are extremely fast and some are very slow. And it also unites many different modeling frameworks like differential equations, Boolean modeling, and flux balance analysis. And it all works under the one hood, so to say. But what you can do, so building a model, it's a fun part. It's a long process, it's a painful process, but the real fun actually starts when you have a functioning model. And what we wanted to do is to see what kind of knowledge we can now generate, what kind of interactions we can learn that were not even known from the single components that we tried to develop previously. And there are two things that I'm thinking they're really relevant for this, and this is damage retention, that the damage that accumulates in the body stays in the body of a mother cell of the yeast while the offspring stay intact. Very important concept in aging that was discovered here in Gothenburg. And then there is a repair. So it's very important that during we age, we need to repair things that are going wrong. And we try to test two different methods or the models that how the organism could repair things. One is a very linear process, and we call this unlimited capacity to repair. And the other one that you look is more hyperbolic. This is the decline in the repair capacity, which simply tells you that you can really push your repair early in life when you reach a peak, and then everything declines in the body, including the capacity to repair. And with having a model, this allows you to simulate thousands of cells in a fraction of time. And this is exactly what we initially did. We simulated the whole yeast population where each of these dots represent one cell. And then we look at the different ages of a mother and different ages of the daughters that they could produce. So we have a whole structured hierarchical population of a yeast. And then we look at different emerging properties like generation time, cell growth, damage at birth, size at birth, and many others. And we look at this in the context of having the present retention mechanism or not, but also looking at these two different repair mechanisms that you can have. So most of the data that we generate are basically trying to see how the retention and the repair are playing the role in us or the yeast aging. So the first thing that we could see is that this retention mechanism is very important to make the larger population. So you can just see by simply looking at the number of dots that we have on each of those, but also makes the whole population more homogeneous. You have this replicative potential, so this how long the yeast lives, and it's extremely high when you have the retention mechanism. And it's very there is a difference between the different repair capacities, but it's not something that can be compared to the no retention case. Then we further we look at the ability to different properties like rejuvenation. Cellular rejuvenation is a very important property, not only for us humans, but for yeast as well, because it maintains the life of the whole population. And we approach this by trying to derive something that we call the rejuvenation index, and tells you how far you can go and how much you can rejuvenate. And if you imagine that you have like a mother cell that would divide 24 times, then its first daughter divides as well 24 times, so there is no rejuvenation happening in this case. However, if you look at the same daughter, but her last daughter dividing only 10 times, then there is a negative rejuvenation because she had produced less offspring than her mother. But then on the other side, if you look at this case, where this daughter produces 24 offsprings compared to its mother that produced only 10, then this means that the second degree rejuvenation has occurred and that we have a positive rejuvenation index. And we try to plot this now across the population and see where are these hubs where the rejuvenation is happening, and how this is correlated both with the retention and repair capacity. And we again can see that the REMID retention promotes the rejuvenation and basically spread it evenly across the population, while in the don't have a retention, you have this very sharp that majority of the population will never rejuvenate or even exhibit negative rejuvenation index, meaning that the population could diminish at some point. The next, what we looked at is how this propagation, when you have the higher damage rates, we see that this is good for you. Like being a bit of stressed, it's usually helpful for aging. So we plotted, like looking at the different sizes of the mother cell, different modes of division, how this correlates with the rejuvenation index. And we could see that under the moderate damage levels here at the beginning, you have very nice rejuvenation property when there is a retention present. That declines the more asymmetrical division becomes. Under the moderate damage rate, the rejuvenation is even more pronounced for the damage retention case, and it reaches basically its peak under the higher damage retention, higher damage rates that the system can go under. So the stress, there is importance of being stressed and being rejuvenated at the same time, how one can age. The next, what we looked, and we were very interested at, inspired by the human aging, is this health span definition. And this is also something that it's really difficult to define. But for us, looking at the yeast, we were trying to think in terms of how we could keep the damage low, and at the same time push yeast cells to divide as much as possible during this period when the damage is under control. And we were dividing, we have also derived the index for the health span, and then basically plotted it against the, again, whole population and look at the response in the individual cells. And this is something very interesting now happening here, that under retention, and if you have an unlimited opportunity to repair your damage, this is actually the worst scenario for you when it comes to your health span. So the health span is extremely low or non-existent. in decline in repair capacity, you have a moderate increase in lifespan across every single individual in the population. So this black and kind of white scenario was something that we definitely didn't expect and that we were very puzzled about. So we looked a bit deeper, so when you have a model, then you could kind of try to find the consequence of this behavior that we see, these emergent properties. So we plotted the health span index across different retention factors to see how this is affected. And we could see that there is this decline in repair capacity that prolongs it. And why this is happening, if you look at one level below, this is because the generation time for those cells is getting a bit longer. So they take a bit longer time to produce the offspring. But at the same time, this ability to increase the generation time allows them to decrease the damage proteins at the birth. So they're born with a lower stress, so to say, and then their ability to have a higher health span in turn gets much, much better. So then the questions that we ask, okay, now we kind of look at those emergent properties, but what is it that we can do? Are there any strategies that we understand that the system is in danger, how we can act and basically improve the lifespan? So these are the typical curves that we are looking. So you have in blue, you have the healthy proteins, and in this greenish yellowish color, we have a buildup of the damaged proteins. And each peak is basically one cellular division that the yeast is going through. And in this case, you can see that the replicative potential, so the yeast cell divided only 5 times. So we looked, okay, what we can do if the cell just continues to divide normally, the damage becomes higher, the replicative potential decreases, eventually killing the cell. But then we looked, okay, what happens if we now, at this point, could make an intervention such that we decrease the damage formation, so basically slow it down. So what will then happen? So it was interesting to see, a bit expected, I have to say as well, from the looking at the equations, then the replicative potential would increase. And then eventually you could, if you slow it even more down, you could have indefinite lifespan in this case. And then we look at what are the costs here. So we have a damage formation, but we have a repair. So let's look at how the repair would look at. So if you increase if you leave the damage repair in a normal rate, that will also reduce the replicative potential. But if you have ability to act on a damage repair level, we could, with a very slight modification, increase the replicative potential dramatically for the yeast cell, and also push it to the very long replicative lifespan. So these are like the balance of what is, now you have to think in terms of also costs. What is it that it's easier to manipulate, and at which point one could do these things. So we look at so yeast is during its life going through so-called metabolic phases. So the yeast is growing, and longer part of its life is spent in the fermentation phase. And fermentation is very familiar to you if you bake. So this is when you're basically the dough is rising, but also if you're producing beer or a wine, that's the phase where actually alcohol is produced. So it's yeast is fermenting there. And then it's after some point when the growth declines, yeast is switching to the respiration phase. And then there is the third phase where there is ethanol is released. So it's the shortest phase of the life. So for this switch between fermentation and respiration, the ATP increase, basically demand for the repair, is the driver for this. So what we were thinking of doing, let's try to push our model and knock out genes so we can see in which phase the yeast reacts the best in terms of prolonging the replicative potential. So we took from the literature most prominent examples where we knew effects, because there is a lot of research on yeast and a lot of labs did these knockouts. And then we would knock them out in a third phase, in a second phase, or on a first phase, and see how this affects the replicative potential and how basically it works with the experiments. Once this was confirmed, we tried to do a lot of perturbations on the enzymes and see what are the pathways and in which phase they are knocked out will influence the replicative potential the most. And we have seen that if you do this smartly, and it means that you pick the right phase to either overexpress or delete an enzyme in yeast, you can manipulate the replicative potential by actually increasing it. And the most important pathways responsible for this oxidative phosphorylation pathway when it comes to the overexpression of enzymes, but when it comes to deletion is glycolysis pops out, but also a bit on the oxidative potential. But these are the two that you could basically pre-select the enzyme, pre-select the phase where you want to knock them, overexpress them or delete them, and then you basically push yeast to divide longer. Then we wanted to go even deeper into this. So we have a list of enzymes that we were testing and knocking out in each of these phases or within the complete lifespan. And what we could see that the phase 2 is the most critical if you would delete an enzyme, regardless of the enzyme, in order to increase your replicative potential. So during this phase, most of the replicative potential will be altered and pushing the yeast to divide more. If you look at the overexpression on the other side, this is kind of all metabolic phases are equally relevant or good or bad, it doesn't really matter. But what is kind of sticking out that the TCA cycle plays a crucial role, and that interventions in the TCA cycle in any of the phases will push the lifespan of yeast even further. So what did we learn from yeast that could potentially be translated to humans? It's very important that you look at the yeast. Also, is not super individual-based society like we are. It's a very much on a population. So we look at this when you invest in yourself. That would mean that investing in repair early in life is extremely important. So the more you can tweak the repair mechanisms becomes better. You should have a fast divisions with a low damage levels. And then when the damage takes over and the repair mechanisms slow down, you should basically stop dividing. This is from the mother's perspective, which will lead to their long health span. And then the other side, if you're an offspring, your mother should keep away damage from yourself as long as possible, which will result in a homogeneous populations where the rejuvenation will be spread evenly across the whole population, resulting in a healthy overall population. But this is yeast, right? So we now know quite a bit of the mechanisms and how this works. So our next step in the lab is basically translating the whole methodology that we have built so far. And this is cool with math, that math doesn't care if you're yeast or a human data. And we are basically rebuilding this in a so-called aging digital twin. The problem that we encounter in the field is that the current practices, regardless of they are coming from the modeling or experiments, cannot predict personalized health or aging trajectories that we would like to see. So what we are now doing, we are leveraging on the machine learning and AI methodologies and combining this with all the modeling that we have done so far in something that it's called the data-informed mechanistic models. And what we are trying to do is basically that you have a health measurements here on the y-axis, and then you have time in a person's life, where we could go to the medical journals, we could identify critical events that the person went in the past, and we use machine learning to find those events, to analyze the data, couple this with the mechanistic models, such that at some point of life, we could design interventions and predict those interventions with so-called hybrid modeling. So you can think that the AI module is basically giving you individual aging trajectory of a healthy data. Mechanistic model will simulate the effects of those. And then what is resulting here in this part is basically a hybrid model that will tell you how the person reacts to the specific intervention based on, for example, their metabolic profile. And this is where we are now in the group. We are putting the math behind all of this and trying to test the pipelines. And we are basically highlighted with the goal that it's a Japanese saying that says that the way that we should age is called pin pin korori. And if I'm not mistaken, that means pin pin is that you should exercise daily. And when your time comes up, you just basically collapse. And that's the best way to finish your life. So it's live long and die quick. It's the strategy that we should all have and equal our life expectancy with our healthy life expectancy. And this work is been done with a lot of people and with a lot of support from the Swedish financial agencies that we are very thankful for. And I would like to thank you for your attention.
Thank you, Marija. This was a very, very interesting talk. Especially, I have a couple of words noted down, and that's it's brutal, the aging definition. It's absolutely fun to discover it, and then die quick. Okay, let me start with a first question for you. Is yeast? Are there certain yeast which are better, which you can model? Let's enlighten the audience, because yeast is sometimes in our series, but not all the time.
Yeah, so yeast that we work with is this called budding yeast, Saccharomyces cerevisiae. That's the most popular yeast that you can work with. And the reason is that it has this asymmetric division. So it's very easy to understand who is the mother, who is an offspring, and how all these things are functioning between. And what is super important, and for me, building the wet lab, you miss all these ethical, basically, permissions and everything if you want to build the experimental lab. So it's very easy to manipulate with. The genetics is known, the experimental tools are known. So it's a simple yeast that you use to bake bread, make wine, and make beer. So it's like 3 key components in life.
I don't ask where you are purchasing them. Okay. Yeah. Then very super interesting, the damage retention. I have to look a little bit, because tell me if I understood it correctly. So the damage retention means that the damage stays with the mother. It's not going to the offspring. Then you have a larger population. They are more homogeneous. I would say then, isn't that of regression to the mean? Is there no evolution because everybody is the same? But you said there is good repair. But if you have a homogeneous larger population, isn't that bad because evolution is not at play?
Yeah, I mean, that's you can. So it's a tricky thing when you look at the yeast wants to optimize its population, right? They want that each individual is contributing for different factors, and this is what we want to do in biotechnology. We want to have a healthy population that could have an impact on a product that we want to get out of the yeast. From the evolutionary perspective, I completely agree with you. This one could understand if this is the best strategy, but we were simply looking at not what is the best, but it's basically from looking at being a yeast, what do you want to even out? And it turns out that this is like this effect of a homogeneous population. If you look at at us, how one could interpret this, it's obviously that we all age in a different rates, and averaging it probably will also not work.
Yeah. So did you stress the population, which was quite big and large and then homogeneous, if they were more stress resilient to certain stressors?
Yes, we look at that. To some, they were more resilient. They show extremely high variability in different stresses. So this is something that we look from if you put it now on the individual level, that you see a very different response. And we have basically methods to look at mechanisms why this is happening. So even if you start from a homogeneous population, the variability in the stress response is very large.
Okay. And could you even then rescue them with repurposed drugs? Grace actually.
Yes.
Okay.
Yes, exactly. That's what I was thinking during her talk. This is exactly what you can do with the methodology. When you identify this like a critical points in someone's life, then this is the good point where you can implement the intervention, and it could be a drug repurposing, supplements, anything that we could act. But the focus here should be on like, let's predict this. That's much faster than or skipping certain parts of the clinical trials and so on.
Yeah. Would love to touch base with you to see how we could shorten that.
Yeah.
So then I thought about elephants I saw in Africa, and they have a long generation time. So you said generation time means better health of the offspring because of better repair, at least in the yeast, if I understood correctly.
Yeah.
So can you translate that to other animal models, like elephants, which have a huge...
There this again, I will stress that I'm a mathematician, right? And my understanding of biology and evolution is probably very, very limited. But there were these theories that the bigger like the systems are, they might live have longer, and what kind of energy expenditure that they have and how they produce. I cannot speculate to translate this to other organisms because definitely I don't have a capacity to it, but I know that there are evolutionary paths that would favor the bigger organisms.
Yeah. And then, of course, it's always the question, is it between the species or is it within the species? And what you did is within the species, you would imagine that if you're over time, then you would be healthier. But let's leave that as a speculation. So I don't know either. Then I was scratching my head because we do lots of human studies, and you do so many experiments, and you have thousands of these mothers and daughters, etc. Do you have to apply for ethics? Is there any ethics involved in working with yeast?
No,
maybe a very naive question, but
no, that's a perfect. I mean, that was my first question when I entered the field. Like, I wanted to build a experimental lab. And the thing was like, okay, let's see what kind of permissions we need. And with yeast, you don't need any of those, so which makes it life extremely easy to work with. So that's, that's the status.
Okay, wow. Because the pathway you were showing are so remarkably also being shown in humans.
Yeah.
So can you tell us a little bit more about the yeast, what they have, what they don't have? We already talked in our webinars about C. elegans, that we can actually mimic sarcopenia or even dementia. So can you tell us what can you mimic and what not?
Yeah, so there was so we selected basically the pathways, particularly those that are like identical in humans. So yeast has 6,000 genes. We have like 24-ish thousand, but 30% of the genes in yeast do exactly the same thing as they would do in humans. And this is particularly relevant for the signaling pathways. So there's a so-called the SNF pathway that it's basically does the same, has the same mechanism that it's in humans. It's an AMPK pathway, and it's first discovered in yeast, and then this research basically translates into the human. So we are basically cherry picking the yeast pathways when we model, knowing that they are translational to the human application. So it's not that we are just like looking whatever. And it's very important to understand that a lot of basic discovery that we know now for ourselves are basically done for yeast, and many of the early Nobel Prizes that will be next week is the big event in Stockholm, are basically for yeast, given for yeast. And we have a guest, it's Paul Nurse, coming here to Gothenburg as well next week. So it's a huge basic research and that contributed to what we know now about ourselves and all these interventions that we can make. So it's extremely exciting organism. You could even do simulations of the Alzheimer on yeast. It's a wild studies that people did. But everything is due to the simplicity, basically, in manipulating it experimentally.
So next time, if our listeners are drinking a glass of wine or dripping on a beer, so okay,
Yes.
we will
Yes.
really value that. Okay, let's switch gears a little bit to your digital twins, because in the healthy longevity medicine field, it's hugely popular now to say, I have a digital twin, and I can actually say how old I'm going to be and what kind of interventions to take. Tell me a little bit what you can really translate from the yeast now to your models, applying that to metabolism and maybe even in humans. Where are you at the stage that we can really have the digital twin?
Yeah, so the machine learning part, that one I could say it's a simple because that's like a methodology that it's already developed, and we already take a lot of health data and analyze it. And there are a lot of papers coming daily, basically. But the core here is this mechanistic, when you derive the mechanism, why this is happening. And this is important for the predicting outcome. And we are now at the stage where we have a metabolic model of human. That is one module that it's ready. And this model on damage accumulation and the growth, it's also applicable for the mammalian systems. And now it's more adjusting the signaling module. It's very conserved, but still it's not completely fit for the human. So we are now trying to make mechanistic part ready, but we are testing the individual modules in combination with the machine learning to see what we can learn, what kind of mechanism we can do, what kind of intervention, and then basically predict the trajectory. And the outcome is basically that we have all these three modules working together on top of the aging, on top of the machine learning part.
Yeah, and I think nearly everybody in the room would agree metabolism is very important, but why have you chosen metabolism first to get, and what are the next stages you want to actually model and then interlink?
The simple thing is this caloric restriction, right? This is like what, I don't know how many decades now, it turned out to be the most effective way. So we want to really see where this caloric restriction plays role. Could you actually modify? That's why the metabolism comes in place. So like a metabolic response under the, so it's a very simple intervention, right? It's could be very cheap and very controllable to do. So that was the motivation to include metabolism first, and first try to see where this caloric restriction comes from and why it becomes so important. And can we learn something from the past of an individual if there are critical events occurring, diseases, stresses, that could correlate with the future outcome of the caloric restriction, for example.
Yeah, great. Before going to Grace, because there are many questions already, I have my last question. And what would you advise youngsters who are now listening, young by heart, by the way, no ageism, if they want to enter the longevity field, the aging field, what would you give in terms of advice?
Yeah, you go for it is the first one. We need more power and try to be open, understanding that there are different fields contributing to the advancing the whole healthy longevity, and don't stick to one method. Try to basically be holistic, as the aging is holistic, so that you have a whole package. And the field is super exciting. The community is extremely welcoming, and I think everyone will enjoy the ride.
One last thing, what makes you most excited at this moment in time?
The development of this data that we have and how we can leverage this data. I'm really excited to understand what is it that we can learn from all of this.
Perfect. Grace, over to you.
Okay, so hi, Professor Marija. Thank you so much for the presentation. We have a few questions about the use of mathematical models to predict aging. So let's start with this one. So, which level of modeling in your historical timeline is currently most useful for finding causal or clinically actionable targets to extend lifespan?
I will say that all of them are extremely important because they build on a different mathematical formulations and different level of description. And sometimes for some processes, we need to lower down the mathematics because we can easily work with that. Some processes we can basically analyze with a very simple math that is on a large scale, and for some processes we need a bit more of a complexity. But there are also this, you can black box things with the math. So I would say that they are all equally relevant, and the knowledge was built in centuries in research.
So you would say like a combination of each would be good.
Yeah, exactly.
Okay. So the next question is more specific to your mathematical model. So can you quantify how much retention contributes to rejuvenation relative to the repair rate?
Yes, we have a numbers. The thing is that you can't program the yeast cell for that exact number. But we have like ratios that are relevant for the tension and in combination with the repair. Yeah.
Okay. So next question is, how conserved is yeast damage segregation in higher organisms? So what do you think are the necessary steps or translational pipeline to move from yeast-based insights to predictions about human health span or longevity?
Yeah, so the damage accumulation is one of the most conserved pathways through the evolution, and it goes from E. coli to yeast to flies to mouse to everyone. So this is one of the most conserved aging kind of markers that you can. So the accumulation of damage, or this damage carbonylation, that it's also known as the process, it's shown for decades now that it's evolutionary the most conserved aging marker.
So we also have a question. It says, what's the biggest challenge when you try to translate these mathematical rules from a single yeast cell to a complex human being?
I would say it's a data. The math, the equations themselves are that's the beauty of math, and what attracted me from the beginning is that it's a universal language. Once you build it, it doesn't care where the data comes from, but the data are the bottleneck. And this is where we have to adjust our equations based on the human data that we are getting and how detailed they are, what is the kind of variability within the data that exists. Yeast is very easy to measure and have time scales that are extremely tight. In humans, this is sometimes not possible. So you have a lot of interruptions in the data that you have to reconstruct. So based on the data that we look at, we adjust then our equations. But they are in essence, they are the same. It's the process that it's called parameter inference that comes on top of it, how much we can learn more about the model based on the data that we have. So I would say that the modeling structure, it's not altered, but it's more the process of learning from the data. It's a bit different.
So today you also talked about your model, which is based on one hallmark of aging, which is genomic instability, and you focus on the relationship between DNA damage and repair. So which hallmark would you be most looking forward to investigate next? Perhaps to come up with a more comprehensive, mathematical model of aging?
Yeah, that's a really, really good question. We just look at the hallmarks the other day in the group and trying to see. So like this, I would say this theoretical background pushes us that we want to see them as a whole. And try to incorporate as many of them as possible, because I think that the like a relationship between them are more important than what they individually do. And this is the part that we are lacking and what we can do mathematically. So I would not favor any particular, but my goal would be that we incorporate basically the whole system.
Yeah, because all of the hallmarks are kind of interconnected, right?
Exactly, yeah.
So we also have a question from someone. They said that you're finding that less repair can sometimes lead to a longer lifespan. Kind of sounds a bit counterintuitive. So could you please explain that in simpler terms? Is it because the cell stops wasting energy trying to fix things that are too broken?
Yes, yeah, that's a very counterintuitive. I have to also agree with that. But it's like if you think that repairing things early on, you have a lot of resources and energy to invest, and then you basically maybe even overdo it. but then you basically allow other processes to work continuously and without disruption later on, and that will kind of help you in the longevity, in the replicative potential.
Okay. So someone also really liked the analogy of the race car sensors. So in your vision of the aging digital twin, what do you think is the biggest missing sensor or data point right now? Is it something in our genes or more about lifestyle data?
That's a really good question. What is another sensor that we can put on ourselves? And I think that it's very interesting about this wearable electronics that you can have that would have more on the. Again, I will go back to this crosstalk between them. If there is a wearable electronic that could synchronize between different measurements, I think that we measure pretty much like not everything probably, but a very big part of it right now. But again, the combinations are, if you have a sensors that could interact with each other, that would be interesting to see.
We also have a question. It says, before translating some of these findings from yeast aging to human aging, are there intermediate organisms where the ideas could most usefully be explored?
I think the mouse is the most natural step. But I think that the way that the field is developing now, that we need to make this higher jump, I would say. And it's something that it will be directly relevant. And I think that at this time we have matured enough when it comes to the quality of the data that we are producing, but also in terms of computational power, machine learning methods, mathematics behind it, and that we are as humans ready to jump directly to the human aging, that we don't really necessarily need intermediate organisms. And I think that's the beauty of having this digital twins, that you could test everything basically in silico first.
So, Professor Marija, I also remember you spoke at the Global Conference on Geophysics earlier this year, where we talked about how this new concept of combining physics, math modeling, and aging biology. So maybe just to end it off, how can we make other researchers appreciate the importance of math in aging biology more?
Yeah, I mean, just think about the amount of data that we have and how much, like, our human brain can work with the 7 interactions at once, and we have way more. Math is a universal language. It's a very cool tool to have, regardless of what you will do. If you are interested in aging, this is probably the best marriage that you can have. So it's I think it's a great time to be alive at and doing research in this geophysics that we coined the term in Singapore in March. And I'm really looking forward that we explore even more methods and even bring more physical models into it and see how we can move forward.
Thank you so much, Marija. I see, and I also got an app from Professor Suresh Ratan, you might know, from Denmark. Why have you chosen the title of your presentation?
How I choose the title?
Yes.
Yes, I have this from my head for like years. When I was starting to do this, I realized that it's this definitely in aging. It's a thing, you know. This being stressed. Grace, you also say in your work at the beginning, it's good for being but the dosing the stress such that it doesn't kill you is probably the best balance. So it's
okay.
Yeah.
Thank you so much. And I think it's even a song, if I remember correctly. So that's great. Thank you so much for being here and really sharing your thoughts and your knowledge. I have a couple of announcements. We organize the Healthy Longevity Intensive course designed for healthcare professionals. It's CME accredited. And if you want to learn more about precision geomedicine or healthy longevity medicine, this might be your course. And this is going to happen from the 23rd to the 25th of February next year, 2026, at the University of Singapore. And this is not the only one. 2 days after the course, we have the Unlock Healthy Longevity Conference, focusing solely on supplements and repurposed drugs. What could we actually give in healthy longevity medicine? And so this is at the 26th and the 27th of February. You might also want to know that there is, it's still open to submit abstracts for the conference. So please do so. There is a total prize of 5,000 Singapore dollar to win for the best abstracts. We also focus on startups because we have to evolve our field. So startups are very important. So we have a pitch competition at the conference, also with a very nice prize of 5,000 Singapore dollar. And it's all happening at the Unlock Healthy Longevity Conference at the 26th and the 27th in February here in Singapore at the National University of Singapore. We have a next Healthy Longevity webinar, and that's happening at the 18th of December 2025, so a little bit before Christmas, with Professor Carsten Weiland. For now, we are closing the webinar and give you a little bit of a teaser of the next episode. Take care.
You're probably our first member from the nursing department. Could you tell me what about nursing that interests you?
Well, I think nursing is both an art and a science. For nursing, we really look at healing not just the physical body, but at the heart as well. Right. What's the thing? I hear black, green, red. Three colors. Green. Final answer, green. What makes me decide to do research is that